Good day, everyone, and welcome to today's EDF Osprey Legacy Society webinar. At this time, all participants are in a listen-only mode. Later, you will have the opportunity to ask questions during the question and answer session. You may register to ask a question at any time by pressing the star and 1 on your touchtone phone. Please note this call may be recorded, and it is now my pleasure to turn the call over to Mo Pepin, Manager of Planet Giving Marketing and Stewardship. Please go ahead.
Welcome everyone. Thank you for joining us for this special event brought to you by the Osprey Legacy Society. And thank you to our Osprey members, those of you who have remembered EDF in your estate plans, trusts, IRA distributions, and gift annuities. Your support for EDF's work in the future is so important for future generations, and your impact is transformational. If you're interested in learning more about becoming a member of the Osprey Legacy Society to enjoy more events like this one and a variety of other benefits, you can reach us anytime toll-free at 1-877-677-7397 or legacy@edf.org. We have a special program for you today on the risks, benefits, and future of genetic engineering and synthetic biology, and it's my pleasure to introduce you to our presenter today. Lisa Moore is a senior scientist in EDF's Office of the Chief Scientist. She began as a Locay Fellow in the Climate and Air Program with EDF after completing her PhD at Stanford University, where she studied the effects of climate change on ecosystems. She spent 3 years on the EDF climate team explaining the causes and environmental effects of climate change and providing scientific advice related to domestic climate policy and international climate agreements. Lisa then joined the Office of the Chief Scientist 10 years ago, where her main focus now is exploring emerging issues that may be relevant to EDF's work to make sure that we stay at the leading edge in all our program areas. One part of her role is organizing a Science Day Symposium twice a year, which is the origin of the content that we will be covering today. So with that, I'll turn it over to Lisa.
Thanks, Mo, and welcome everyone. I'm really glad to have you here today. As Mo said, I'm a senior scientist in the Office of the Chief Scientist, and today I'm going to present a condensed version of our most recent Science Day, and then I look forward to answering your questions. Science Day is a symposium that we organize twice a year as part of a Board of Trustees meeting, and the goal of Science Day is to explore an issue that may be relevant to our mission. We don't make strategic decisions at these events. Instead, we use the time to learn the latest science from leading researchers. Our latest Science Day focused on understanding the risks and benefits of redesigning nature using new genetic engineering and synthetic biology tools. Today, I'm going to start by introducing some of the biotechnologies that we talked about. Then, I'm going to summarize some potential applications of these technologies for protecting biodiversity and public health. I'll also briefly describe the regulatory landscape and recommendations that experts are making for improvements. And then I'll wrap up with some take-home messages. But actually, before I start, I want to give you a sneak peek at those take-home messages to help frame what I'm going to talk about. These new tools are extremely powerful, and they're advancing very rapidly. Researchers are thinking about using these tools outside the lab to address environmental and public health challenges. And all of these potential applications need to be evaluated and regulated with a heavy dose of risk assessment and management, but it's important to consider the enormous potential benefits too. These technologies may provide positive solutions to otherwise intractable challenges, and they could help build resilience in natural and social systems. However, regulators are completely unprepared to deal with these new technologies, so we need new governance structures. And finally, there's been a lot of engagement by NGOs and others on GMOs, especially food products developed using older biotechnologies. But most of what we're talking about here are completely new tools and applications that are meant to be released into the natural environment to solve environmental problems. Very few conservationists right now are involved in discussions and decision-making around these tools. So this Science Day was a really important opportunity for us to start thinking about the issues and how we might engage on them. So what kinds of technologies are we talking about? Most of us have heard about genetic engineering and gene editing. Genetic engineering generally refers to the transfer of genes from one species to another. Gene editing generally refers to copying, cutting, and pasting pieces of DNA within the same species so there's no mixing of species. And then there's synthetic biology, which is a rapidly emerging field that goes way beyond these earlier biotechnologies. It uses computing and engineering approaches to design and construct completely novel organisms and biological systems using material from the lab rather than from organisms. As a result, synthetic biology can do things that aren't possible with older biotechnologies. Our first speaker describes synthetic biologists as using a familiar concept from problem solving and engineering, and that is a design, build, test, and learn cycle. But here, instead of using hardware, the scientists are using biological materials and techniques. One thing that was clear from Science Day is that this is not science fiction that's years away. Instead, kids are actually doing it today. This is a picture of the teams competing at last year's International Genetically Engineered Machines competition. Most participants are undergraduates, but there is a growing division for high school students, and these people come from all over the world. There are also thousands more people who are experimenting with synthetic biology in community labs that are popping up around the world. At Science Day, we heard about 5 potential or planned applications of these biotechnologies for protecting biodiversity and public health. 2 of them are being developed to detect and respond to environmental threats like pollution and warming, and 3 are meant to address biological threats like disease and invasive species. And as you'll see, the applications are at different stages of development. A couple are still in very early stages, but 2 are actually starting field trials. Our first speaker designs plants that can detect specific pollutants. If you know the molecular structure of a pollutant, she can design a specific sensor gene on a computer, then synthesize the gene in the lab, and encode it into the plant's DNA. She compared this computational protein design to building integrated electronic circuits, except that she's using genetic and biological material. She's designed plants that change color when they're exposed to specific chemicals, like explosives. For example, here's one of her plants before being sprayed with the chemical it was designed to detect. When it's exposed to the chemical, it turns white, and then when the pollutant is removed, the plant resets to green. Right now, her research is only in the lab, but she envisions using these plant detectors in places like near chemical plants or in airports, or even in public parks or along streams. Our second speaker focused on research to engineer heat-tolerant corals. Corals are animals that host a photosynthetic algae. The corals provide the algae a place to live, and the algae provide corals 90% of their nutrition, and they also give corals their color. When corals get too hot, they expel their algae and they turn white. This is called coral bleaching. And if the coral hosts aren't recolonized by algae quickly enough, they starve to death. The oceans are warming very rapidly, and coral bleaching has reached such a crisis point that scientists are trying to develop genetic engineering approaches to save coral reefs. Corals can host different kinds of algae, and the strain of algae that affects the host's heat tolerance. This photo shows 3 corals of the same species in the same environment after a marine heat wave. These 2, which did not bleach, host the same strain of algae. The one that did bleach had a different strain. To be able to design heat-tolerant corals, then, scientists need to understand the genetic drivers of heat tolerance in both organisms. For example, scientists have developed different strains of algae that are heat tolerant on their own. This next graph, with all of the colors, shows expression levels of stress response genes in those 3 heat-tolerant strains of algae during heat stress. The red colors indicate higher levels of gene expression, and blue colors represent lower levels of expression. You can see that gene expression differs among the strains, even though they're all heat-tolerant. So what happens if you put these heat-tolerant algae into coral hosts? Scientists found that only one of the strains conferred heat tolerance on its host. The other 2 host corals were still vulnerable to heat stress. Then they looked at the coral host's gene expression levels during heat stress, and they found that gene expression was affected by which strain of algae was living in the host. So now scientists hope that the information from these sorts of experiments will help them identify promising genetic targets for engineering heat-tolerant corals. Our next speaker talked about how genetic engineering can be used to develop blight-resistant American chestnut trees. The American chestnut was once a dominant species in the eastern United States, In fact, it was sometimes called the redwood of the East. It had tremendous cultural and ecological importance. It provided food and shelter for people and wildlife, and it supported the region's timber industry. In the early 1900s, a fungal disease called Chestnut Blight was introduced from Asia. The fungus enters wounds on trees and it produces oxalic acid, which kills the tissue under the bark. American chestnut trees were devastated by the blight. Today, scientists are trying to restore them using both breeding and genetic engineering. Scientists have had some success with crossing American chestnuts with a Chinese species that is resistant to the blight. But breeding takes time, and it isn't very precise because resistance and inheritance are complicated. So now researchers have turned to genetic engineering. The current genetic engineering approach inserts a gene taken from wheat, which detoxifies the oxalic acid produced by the fungus. This increases the tree's resistance to the blight. This method is faster and it's more precise than traditional breeding. The scientists are now testing for environmental or health risks from the transgenic trees. For example, is the nutrition of the nuts different? Will the pollen have adverse effects on insects? Does the transgenic leaf litter affect native plant growth? So far they've done these and many other tests, and they have not found any enhanced risks. Now the scientists are planting transgenic trees in large outdoor plots for long-term ecological research. Ultimately, their goal is to have members of the public plant the blight-resistant trees throughout the chestnut's original range, but first they need to obtain permits from the agencies that regulate the GMOs. The scientists are also doing additional research to identify other genetic mechanisms for blight resistance. Our next speaker was an expert on genetic control of malaria mosquitoes. Malaria is spread by mosquitoes, and it kills about 400,000 people each year in Africa alone, and 2/3 of them are children under 5. Public health officials have had great success in reducing malaria deaths over the last 20 years by using control methods like insecticides and bed nets, but the results have started to plateau. Now, scientists and public health officials are considering genetic control measures that would let the insects themselves do the work. For example, you could engineer mosquitoes that are sterile, which would suppress or eliminate the population. Or you could engineer mosquitoes that are resistant to the malaria parasite to replace the current mosquitoes with the new ones. This approach would be species-specific and non-toxic. But the problem is, how do you spread a trait throughout a whole population? Scientists are experimenting with a tool that's called gene drive. If you think back to basic genetics, you learned that a given gene has a 50% chance of being passed from parent to offspring. But a gene drive is different. It can bias its own inheritance. That means that if you introduce it into a population, with each generation, the trait becomes more and more frequent in the population until every individual has that trait. Scientists have succeeded in developing gene drive in malaria discrete mosquitoes. Last year in a lab experiment, they introduced a sterility gene drive, and they were able to eliminate an entire population of malaria mosquitoes in as little as 8 generations. This graph shows egg output in a population of caged mosquitoes, and egg output went to 0 in just a few generations after they added a few individuals carrying the gene drive. However, implementing a gene drive system in the real world requires more than just molecular biology. That's why researchers are working with a large collaboration of citizens and public health officials to address questions like ecological effects, containment, regulation, and community acceptance. They're now planning small-scale field tests in Africa. Another potential application of gene drive is to control invasive species on islands. In the Galapagos, the Pinzon tortoise has not successfully raised any young in the wild in the last 150 years. That's because invasive rodents eat their eggs and their hatchlings. Only captive breeding has kept this species from going extinct. The tortoise's story isn't unique. 75% of all extinctions in the last 500 years have occurred on islands. 86% of all recorded extinctions on islands are because of invasive species. And it turns out that 88% of all threatened species on islands are impacted by just 4 species of invasive rodents. The reason that rodents are such a threat is that they eat seeds, eggs, and baby animals. So, if you want to prevent extinctions, you should be working to remove invasive species from islands. And rodents are a very important target. Right now, invasive rodent control means dumping rat poison from helicopters. But that can't be used everywhere, it doesn't always get all of the rats, and it can potentially endanger native species. That's why some conservationists are starting to look at genetic control as a way to let the rodents do the work, just like we saw for mosquitoes. Researchers at several different universities are developing gene drives that would eliminate a rodent population within a few generations. Now, this is just theoretical at this point, and they are a long way from even doing lab tests. But the effects of successful rodent control are phenomenal, and that's why some island conservationists are considering this as a potential new tool. This is Palmyra Atoll. Invasive rodents were eliminated from the island in 2011. When invasive rodents were no longer eating plant seeds, the native plants rebounded within just a few years, and now the ecosystem is thriving. And you remember the tortoise I told you about earlier? Conservationists eliminated invasive rats from that island in 2013, and they recently found the very first tortoise hatchlings to survive in the wild in 150 years. Now, in all of the examples I've shown you, there are unknowns and risks to be weighed against the potential benefits. How do we make those decisions about developing and using these emerging technologies? Who makes those decisions, and what values and worldviews should drive those decisions? Our final speaker made it clear that our current regulatory framework is completely inadequate. Just as an example, in the United States, the current regulatory framework, the Coordinated Framework for the Regulation of Biotechnology Products, was established in 1986, and it has barely been updated since then. 3 different agencies have some jurisdiction over most genetically engineered organisms, and the rules for which kinds of organisms fall under which agency's jurisdiction aren't always clear. And some organisms that don't fit neatly under any agency's jurisdiction are exempt. It will only get worse with the new technologies and products that are coming on board, and of course, that's just in the United States. The picture gets even more complicated when you look at regulatory frameworks around the world. We also need to acknowledge and consider different values and worldviews. Our failure to do that thus far has led to a lack of trust. Our speaker and her colleagues have been working to develop recommendations for new, flexible frameworks for governance structures that can handle new technologies and products like gene drives. These governance structures need to be able to deal with different levels of certainty, familiarity, complexity, and they need to respect different values and worldviews while being informed by science. So what were the key take-home messages from the symposium? First, synthetic biology and gene editing are here, and they are advancing rapidly. Second, these techniques have powerful implications for conservation and public health. It's easy to focus on risks and unknowns, but it's important to keep the benefits in mind too. These are potentially transformative solutions for protecting biodiversity and increasing ecosystem resilience in the face of environmental crises. Third, our current regulatory structure is woefully unprepared to deal with these new biotechnologies and their potential applications. However, researchers have made recommendations that policymakers could consider. And finally, an issue that came up several times was that few conservation groups are engaging on these new tools and their potential applications. The scientists we spoke with really wanted more NGO voices at the table. So, condensing about 5 hours of material into 15 minutes leaves out quite a lot, but fortunately we have time to dive into more detail, and I look forward to taking your questions. Operator, could we have the first question, please?
At this time, if you would like to ask a question, please press star and 1 on your touch-tone phone. You may withdraw yourself from the question queue at any time by pressing the pound key. Once again, to ask a question, please press star 1 now. And our first question comes from Amira Mansour. Please go ahead.
Hi, I'm wondering, is there a way to get a link for those slides? It's fascinating, and I just can't take notes fast enough.
Thank you. Yes, we'll be able to get this information to you, and when we wrap up, Mo will be talking about ways to contact us, and we'll be sure to reach out and share this with you. Thank you for your interest.
Yeah.
Operator, can we have the next question, please?
Our next question comes from Brittany Henderson. Please go ahead.
Hi, my question is about unintended consequences. What are the scientists doing to kind of mitigate the law of unintended consequences with this genetic engineering? Because, you know, if they're, for instance, setting loose a GMO population of a certain species, you know, how are they making sure that doesn't become invasive and kill off biodiversity or, you know, some other harmful effects?
That's a great question. Thank you. I think that the scientists who are developing these applications really want more voices for precisely those reasons. They want more ecologists, more conservationists, more environmentalists at the development stage to even raise those questions that they may not be able to think about. But it's a huge challenge because some of the testing has to include real-world implementation at small-scale field trials, right? And so to collect the data that you need, yes, it's a difficult thing. I think having more environmentalists, ecologists, public health officials, all sorts of voices, whether they're from science or affected stakeholders, need to be in this discussion right now to raise those questions, to think about how to study those, to check for, be aware of, monitor for those sorts of unintended consequences. So it's certainly something that's top of mind and a reason that we want, you know, why they want more people involved. Thank you for your question. Operator, can we have the next question, please?
Certainly. Once again, if you would like to ask a question, please press star and 1 on your touch-tone phone. And our next question comes from Joan Wills. Please go ahead.
Thank you. What's being done and what processes are being used to create a knowledge base to change the governance structures, the regulatory governance structures?
Great question. Thank you. Right now, it seems to be mostly in the academic world. There have been reports, for example, by the National Academy of Sciences on the future of biotechnologies and governance structures for them. Our closing speaker at Science Day was one of the leading thinkers on this, and she has written several reports digging into these new technologies and suggesting ways to, you know, collect the knowledge base, as you say, for shaping the governance structures, as well as flexible frameworks that can deal with that uncertainty and those unknowns. But as far as we're aware right now, there isn't any actual language that could fit into the existing regulatory frameworks of the various agencies so far. So I think that's a gap that needs to be addressed. So the researchers are on it. They're making recommendations. The next step is to push that into the policy realm and try to develop, you know, something solid that goes into the regulatory and governance frameworks. Thank you for raising that question. Operator, may we have the next question, please?
Our next question comes from Nancy Halbert. Please go ahead.
Yes, I just wanted to ask a question about public perception of, you know, GMOs or just editing genes, etc., and how, you know, how that plays into the whole picture of what you're doing. Public sphere. I mean, the public sphere of it.
Yes, thank you. I think one of the things that we learned in putting the Science Day together was just how far along the science is. And I think that there is a gap in knowledge and understanding and just awareness of the existence of some of these tools in the public sphere. And I have not looked at any studies on that. I may be wrong that there are obviously some of the high school students and the undergraduates who are playing around in these community biolabs or competing in those international competitions are aware of it, but I don't know that there's that much awareness or engagement by the public. Certainly the NGOs are not that engaged at the moment. except for, as I said, some of the earlier biotechnologies. I can tell you that EDF's participation in this field right now, we have an online position statement on biotechnology in general, and we can share that link with folks who are interested. And one of my colleagues is participating in a couple of efforts associated with gene editing in food with an informal NGO network. But again, the public sphere is incredibly important because of the values and worldviews that those different people in the public carry. And one of the nice things about these flexible frameworks for governance that our final speaker and her colleagues were talking about is opening up space to have more of those conversations, to bring in affected stakeholders and the public into those discussions from the very beginning, to lead to the regulations that are finally put in place. So I really appreciate that the academics and the folks who are making recommendations are really trying to build that in, because the public sphere is so important.
Oh, good. Thank you.
Mm-hmm. Thank you for your question. Operator, could we have the next question, please?
Our next question comes from Jonathan McClure. Please go ahead.
Hi, this is actually Catherine McClure. I have a question about what lessons we might have learned from the technology with the CRISPR technique that has been developed and deployed with in helping to control the Zika virus.
Great question. Thank you. We didn't talk about that specifically, so I'm afraid I won't be able to give as detailed an answer as you're hoping for. And so one thing that I can do two things. One is to talk about a related topic that did come up during Science Day, and then I can also follow up with more details offline. Our speaker who talked about governance structures gave a case study of a genetically engineered mosquito that a private company had developed to try to reduce dengue fever, and they planned to release it in Florida. They got permits, they got approval from various agencies, but then the community voted it down, and so it was not released there. It was eventually, because the regulations are so complicated, they were able to change the labeling on the mosquito to move to a different agency and a different regulatory structure and try it under a different name, essentially. And it really raised a lot of concerns about conflicts and differences between how people interpret scientific uncertainty and what counts as where's the line for precautionary principle. So she showed some of the data that came from some of the monitoring of that trial and how different groups interpreted it and what it meant for regulation and decision making. So I think there's a lot of lessons to be learned there about the importance of talking to all of the affected stakeholders and talking about up front how you're going to interpret the data and uncertainty. What's your threshold and why are you choosing that? But I can certainly look into more about and talk with my colleagues about CRISPR and Zika in particular. And my colleagues are on the call are keeping track of the questions that people are asking so that we can follow up offline. I hope that's helpful. Operator, can we have the next question, please?
Certainly. Just once again, as a reminder, it is star and 1 to join the question queue. Our next question comes from Michael Heffler. Please go ahead.
Thank you, and thank you so much for this presentation. It's really been quite fascinating. Given all the things you've covered here, I was wondering, what are you most excited by in terms of a prospect, and what are you most concerned with?
Oh, thank you. Gosh, I can't speak for a whole organization. I think, an obvious way for NGOs and others, and potentially EDF, but again, Science Day is not for making strategic decisions. It's it's about learning, and I can say that this Science Day has catalyzed some internal discussions about learning more, about thinking about where we might act. Do we need to revisit our biotechnology position statement? It didn't really deal with synthetic biology, etc. Something that definitely jumps to the top of most people's lists as something to consider is pushing policymakers and regulatory agencies to try to revamp this framework to make more sense and to be just improve it, rather than building on something that's from 1986 that was envisioning just a completely different set of tools and products. Try to improve the way that we do governance, regulation, decision-making, engagement with different stakeholders. And I think EDF and other organizations could be very well suited and well positioned to make that link between the academics and the researchers who are thinking about this and the policymakers and the agencies who would have to actually write it and implement it. So again, we haven't made any decisions, but I think that could be a potential role for us to play. As far as what most excited us, gosh, I think everybody was really. I think one of the things that jumped out for me and for many people that I've spoken with after the fact was it's so easy to think about the unintended consequences and the risks. And I'm an ecologist. I think of so many risks, and because systems are complicated, you tweak one thing and there's ripple effects, and you know, you can't necessarily model that, and that's scary. On the other hand, so many of our speakers were saying, I don't want to be engineering corals. I want to be able to do something less invasive. You know, what I really want is for the world to reduce greenhouse gas emissions so that we don't have coral bleaching in the first place. But I care about these systems, and this is an important tool. And when Carl Campbell, who talked about rodents on islands, he doesn't want, he's looking at this with cautious optimism, because extinctions are such a crisis, and it is so hard and in some places impossible to go after some of these invasive species, and the results of success are just phenomenal. So I think something that jumped out for people was the benefits that the, you know, not using these is a decision too that does also have impact. So I think that was eye-opening and thought-provoking for all of us to really see some of the benefits that we have seen or that they anticipate. So I hope that gets at your question, and thank you very much for raising it. Operator, could we have the next question, please?
Yes, our next question looks like a follow-up from Amira Mansour.
Yes.
Please go ahead.
It's actually not a follow-up on my original question, but as I listen to this, and I'm not a scientist by background, I'm just wondering how, besides, you know, general support to EDF and related organizations, how does the non-scientist get involved in, maybe it's PR issues or helping on these discussions on ethics or whatever? How can we, as just general populace that's not necessarily science background, help in this, you know, any of the work that you're doing?
Mm-hmm.
Yes.
Great question, and a difficult one to answer. But I can suggest a few things. There are groups that are trying. For example, two of our speakers came from North Carolina State University, which has a whole center focused on genetic engineering governance, how the public, you know, intersects with the science and policy. They've recently, I'm not sure if it's open yet, but soon or very recently, they are launching an exhibit on campus that brought together scientists and artists to think about speculative futures of how these technologies might be used. And it's a, you know, public display of these art installations that envision how synthetic biology might be applied for, you know, protest and some really interesting ideas. And I thought it's really great that they're doing that because, for the folks who are there in North Carolina, they can talk to the researchers, they can think about what the potential applications might be. I would hope that sort of thing would become, it would be lovely to see something like that as a traveling exhibit or something that happens more broadly. I can certainly reach out to the folks at North Carolina State who helped put that together to ask them what other fora might be coming up. You can also look into the IGEM, which is the International Genetically Engineered Machines Competition. It's open. They have a great website. I believe it's igem.org, but I can double check and get back with you. And then, so those are two ways to do it, and I'll keep pondering that, and I'm sure that some things will occur to me later, and we can reach out with additional ideas. But I'm so glad that you want to become engaged, and just learning, asking questions, talking with scientists, talking with elected officials and regulators, and just raising voices, I think is really important. So I'm sorry that I don't have a hard and fast list of things you can do, but there's some edges, some toeholds there, and I'll try to work on that a little bit offline. Thank you. Operator, could we have the next question, please?
Once again, that is star and 1 to join the question queue. And we will pause a moment to allow questions to join the queue. And our next question comes from Patrick Danazer. Please go ahead.
Hey, I'm wondering about how this research is funded and coordinated. It seems like it's a bunch of dispersed academic labs working on separate angles on the problem. So I guess really two questions. One, is there a sufficient effort to fund all this research through NSF or whichever body? And two, is there any effort to coordinate this research, or is it all kind of a cottage industry at this point?
Great question. So each of these researchers will be, most of them were academics, which means that they're generally going to be getting agency money like NSF. Island Conservation, which is the group looking cautiously at gene drive for rodent control on islands, is working in collaboration with some universities and other research institutions. The gentleman who joined us from Australia works for a national research organization that has national funding for the research on coral reefs. So there's, like most research, academics are cobbling together funding from a variety of sources. I would say that probably 5 of our 7 speakers already knew each other, so these folks talk to each other in the research area. Several of them were involved in a recent report calling on conservation groups to become more involved and talking about, highlighting some potential conservation applications for these new technologies. So they're certainly talking to each other, but as far as one coordinated central funding source, I'm not aware of one. One thing I can certainly go back into their original slides to look at their acknowledgement pages, where they always list their long list of sources, and EPA, NSF are going to show up often, and then there will be other smaller grants. I hope that answered your question. Operator, could we have the next question, please?
Certainly, and as a reminder, that is star and 1 to join the question queue. Our next question comes from Leah Landsman. Please go ahead.
Hi, what is EDF doing in relation to these ideas and issues exactly?
Great question, thank you. And I knew this one was going to come up. And, Science Day is not meant for strategic decision making, and we choose topics that we're not working directly on. However, we have been tracking this in a few ways. So I can tell you a few things. Like I mentioned earlier, we have an online position statement on biotechnology, and we can share that link. I've been following, I and several of my colleagues in a variety of different programs, we've been following different aspects of what are called innovative food products. That might be lab-grown meat, some of the plant-based meat replacements. Most of those do not use genetic methods, but some of them are looking into it, and so we've been paying attention to that research and thinking about what sorts of environmental and socioeconomic consequences those products might have, as well as safety and regulation. We have been following some of the literature and the recent National Academies of Science report on the on these new technologies and governance structures. And then my colleague, Doria Gordon, who's a lead senior scientist in both the Office of the Chief Scientist and in the Ecosystems program. I think I mentioned earlier, she's a member of an informal NGO network that focuses on environmental issues and food, and they've been talking about gene-edited crops and where and how those might be deployed. And really our only action so far on that informal NGO network has been to comment on some proposed regulation by USDA on gene-edited plants. She's also involved in a pilot project on cooperative governance of crop gene editing, specifically applied to crops that provide continuous cover in ag production systems, and that's being run out of the University of Minnesota. And we're one of multiple stakeholders that have participated in a workshop and in interviews so that investigators there can understand the different perspectives on the use of gene-edited plants in agriculture. So overall, we're trying to track developments in the field, but we don't have any active projects. But as I said, Science Day certainly really generated a buzz among our staff and our trustees, and there have been follow-up conversations. I haven't attended all of those, so I don't know where they're going, but there may be something coming, and we'll see in the coming weeks and months what happens. And as I said earlier, I would think that one potential area for us to be active would be to try to improve on existing governance and risk assessment frameworks. Thank you for your question. Operator, can we have the next question, please?
And once again, that is *1 to join the question queue. We will pause a moment to allow questions to join. And it does appear there are no further questions over the phone at this time. I'd like to go ahead and turn it back to the speakers for any closing remarks.
Okay, well, thank you very much. I want to thank everyone for joining us today. And I work in our San Francisco office. I want to reach out especially to those on the phone who are affected by fires here in California. I hope that you and your loved ones are safe from the fire, the smoke, and the power shutoffs. And we're all hoping that this will resolve soon. So I do hope that you're all safe and will remain so. I want to thank everyone again so much for your continued support. We couldn't do our work without you, and we're really grateful to have you as part of the EDF family. So now I'm going to hand it back over to Mo to wrap up with some closing information, and thank you again.
Thank you, Lisa, and thank you again, everyone, for joining us today. If you have any follow-up questions or would like more information on this content or other Osprey Society happenings, please do contact us at legacy@edf.org or toll-free at 1-877-677-7397. And we will be circulating a recording of this to those who joined us today as well. So for those trying to take notes on your end, rest assured, we'll get back to you. And on behalf of all of us at EDF, just thank you very much for your interest and support. And we look forward to our next installation of this webinar in the spring. Thanks so much.
Does conclude today's program. Thank you for your participation. You may disconnect at any time.